Section 8 of Radioisotopes and Life Processes. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Radioisotopes and Life Processes by Renato Baserga and Walter E. Kizilewski. Protein Synthesis, the Molecules that Make the Difference. If a man will begin in certainties, he shall end in doubts. But if he will be content to begin with doubts, he shall end in certainties. Francis Bacon. Proteins occupy a central position in the structure and functioning of living matter and are intimately connected with all the metabolic reactions that maintain life. Some proteins serve as structural elements of the body, for instance hair, wool, and the scleroproteins of bone and collagen, the latter an important constituent of connective tissue. Other proteins are enzymes, which are extremely important since they regulate all metabolic reactions. Most of the proteins in the tissues of actively functioning organs, such as the liver and the kidney, are enzymes. Other proteins participate in muscular contraction, and still others are hormones or oxygen carriers. Special proteins called histones are associated with gene function, and the antibodies that an organism produces to defend itself from bacteria are also proteins. The differences in proteins, especially in enzymes, account for differences among cells. It is now appropriate to ask what makes one protein different from another. We know that the structure of a protein depends upon several factors, such as the molecular weight, but the main differences among proteins depend upon the sequence or order of the amino acids that are linked together in the protein molecules. Amino acids and protein structure. Amino acids are the fundamental structural units of proteins. There are 20 amino acids found frequently in mammalian proteins, and these molecules may be linked to one another to form a chain called a polypeptide chain. The structure of a protein then depends on 1. The quantity of each amino acid present, 2. The sequence of amino acids in the polypeptide chain, 3. The length of the polypeptide chain, that is, the molecular weight, and 4. The folding and the side nonlinear arrangement of the polypeptide chain molecules, that is, the secondary and tertiary structures. How can we investigate protein synthesis by using radioactive isotopes? Since proteins are made up of amino acids, the logical conclusion, after what we have learned about DNA synthesis and RNA synthesis, is that the best way would be to mark an amino acid and follow its incorporation into a molecule of protein. We could label a mixture of several amino acids, but for the sake of clarity, we will describe the incorporation of a single labeled amino acid. Labeling an amino acid with a radioactive isotope. Suppose we have the amino acid leucine labeled with carbon-14, and we inject a solution containing it into an experimental animal. Since leucine is incorporated into proteins, if we isolate the proteins and determine both the amount of proteins and the amount of radioactivity, we can measure fairly accurately the rate of protein synthesis. Autoradiography, by the way, is of little help in studying most protein synthesis because all cells are always synthesizing proteins, and so are all labeled after a single exposure to a radioactive amino acid. With RNA precursors, autoradiography at least told us where RNA was being made, but with amino acids we do not even get this information because proteins are synthesized both in the nucleus and in the cytoplasm. Under these circumstances, radiochemical methods are better for studying protein synthesis. Proteins are isolated from the residue left after a nucleic acid extraction process similar to that described previously, and the amount of protein is determined by a simple colorimetric analysis based on comparison of the color of the solution with a standard color. The amount of radioactivity, remember that we are now using a precursor labeled with carbon-14, can be determined with a gas flow counter, which is probably more widely used at present than any other instrument for counting beta emitters, chiefly because of its reliability and low cost. End of Section 8.